All right, everybody, let's go through the Unit 2 exam review together. So as a reminder, these are unit reviews, so not everything on the exam will be on the review, not everything on the review will be on the exam, but I test on important things and I teach on important topics, so there is a lot of correlation. All right, let's start with chapter 7, which is on blood. So what are the major components of blood? And of course, you've got all your videos you should be watching to go through the details, but blood is basically going to be split into plasma and formed elements. So it's slightly more than half formed elements. The other half is plasma. So I'll talk about plasma here first. Plasma is the fluid portion of your blood. It's almost all water, about 92% water. But and you're going to have a lot of dissolved things in it, vitamins and minerals, etc. But the key thing for now to understand about what's in plasma is the plasma proteins, and that'll be coming up in a question in a moment. So with the formed elements, let's go ahead and answer the next question, and then we'll go into that. Why are they called formed elements instead of blood cells? So it's not plasma and blood cells, it's plasma and formed elements. That's because not all of your blood cells are actually cells. So the best example of that would be platelets. Platelets are actually cell fragments. They came from a huge cell called a megakaryocyte, which sloughed off, let's say, 2,000 or 3,000 of them. But a platelet is not a cell, it's a cell fragment. Now, you could argue, based on things we learned in Unit 1, that even red blood cells aren't cells, because to be a cell, you have to have DNA, and red blood cells used to, but they actually, red blood cells have to get really small and really flexible, so they actually jettison off, they get rid of their nucleus, where their DNA is, as well as their other organelles. That's why red blood cells are really the only cell type in your body that has to use glucose for fuel, because they can only undergo glycolysis. They don't have mitochondria, so you could even argue that red blood cells aren't cells. So that's why they're called formed elements. It's semantics, but still important to know. So we'll go through the key types of white blood cells actually in a little bit when we get to chapter 9, so I won't walk through all of them now. Just understand that white blood cells play a big role in immunity for now, in 20 minutes or so we'll be there. All right, let's see, what are some of the key roles that the plasma proteins play? So the three big plasma proteins are going to be your albumins, your globulins, and then fibrinogens. We'll start with the easy one. Fibrinogen becomes fibrin when your blood clots. So fibrinogen, think blood clotting. Globulins, the key thing we'll learn about here in this unit is the immunoglobulins. So those are going to be your antibodies, IgA antibodies, for example. So globulins, think your immune system. Now, there are globulins that also transport things. The best example I can think of, top of my head, would be sex hormone binding globulin that actually carries our sex hormones around. So they do play a role in transport. But generally, I think of the immunity aspect the most. But the third plasma protein that I think is the most important are albumins. So albumin, it is a transporter, but its primary function is to be used as a solute to keep water where it's supposed to be. So albumin plays a major role in osmosis and where body fluids are. If you have enough albumin in your blood, then most of the fluid that's filtered out of your blood during the day will be reabsorbed. If you don't have enough albumin, then fluid will be pushed out of your capillaries into the spaces between your cells called interstitial spaces. So it will become interstitial fluid, but it won't be sucked back in. So a couple of key examples there, someone undergoing liver failure, let's say it's cirrhosis, cancer, whatever, they will develop a condition called ascites. ASCITES, where they'll accumulate massive amounts of fluid around their abdomen. My wife, she works in digestive oncology, and they have patients where they have to remove 15 liters of fluid a week off their abdomen. So the fluid is being filtered out like it's supposed to, but it's not being reabsorbed like it's supposed to because of a lack of albumin. Another time you'd see that is if you see a child that's very malnourished in the developing world, they're really skinny, but then they have a pot belly. Well, that is the same thing. It's they actually are so protein malnourished that they can't make enough protein for this to occur. So here in America, if you see it happening, it's because the liver is failing. In the developing world, they're protein malnourished children. So fluid's being filtered out, but there's not enough albumin to bring it back in. So those are the basic functions of the albumins, globulins, and fibrinogen. Next, what is hemoglobin and what mineral gives hemoglobin its affinity for oxygen? So about 95% of the protein in red blood cells is hemoglobin. Hemoglobin is designed to carry oxygen. So now it's really cool. The structure of hemoglobin is almost exactly analogous or the same as the structure of chlorophyll. So hemoglobin is like this energy carrying protein in humans. Chlorophyll is the equivalent energy protein or energy carrying protein in plants. But the difference between the two is the actual minerals. So what mineral gives hemoglobin its affinity for oxygen? That is iron. So you can have perfect hemoglobin. If you don't have iron, you'll still develop anemia. You won't be able to carry oxygen. So hemoglobin is really designed to hold iron. Iron is what has the affinity for oxygen. Now, if you're talking about chlorophyll in plants, it's actually going to be magnesium. That's why food sources of magnesium are generally green leafy vegetables. Okay, how long does the average red blood cell live? So remember, red blood cells, I just mentioned earlier, they don't have organelles anymore. They spit them out. So they don't have centrioles, they don't have a nucleus anymore, so they can't divide. So red blood cells are produced from stem cells in bone marrow, and they only live for give or take 120 days. So that's an average, but that's how long the average red blood cell lives. So they can't live longer. Some white blood cells can live a lot longer because they have organelles, they have the cellular cleanup crews, they can divide, etc. All right, next. What are the key features and uses of each type of white blood cell? When would you see each elevated? So let's we'll start with neutrophils. Neutrophils are the most common white blood cell, about 70% of white blood cells, give or take, and they are really, really good phagocytes, which means they're really good at engulfing things. So neutrophils are going to be elevated during bacterial infections because they can engulf bacteria by the thousands. They really are amazing phagocytes. So they actually can eat and eat and eat until they burst. So a neutrophil will eat bacteria up, or whatever it's eating, usually bacteria, until it blows up. And when it does that, it releases toxins in the environment that kill even more bacteria. So it's very good at killing bacteria. That's really pus. If you think about pus, pus is primarily dead neutrophils and all the bacteria they've taken with them. So that's going to be neutrophils. Next, let's see, we've got the eosinophils. Eosinophils, they are phagocytes as well, but their primary function, or at least the way I see it, is they're going to be elevated during allergic reactions, but also parasitic infections, because they can basically hose parasites down with toxins to kill them that are too large to engulf. So they are phagocytic, but when you think eosinophils, think allergic reactions and parasitic infections. Next, let's see, we have the basophils. Basophils are the white blood cell that release histamine and heparin. So they're involved in the inflammatory reaction. So if your basophils are elevated, you know you have a ton of inflammation. There's another cell called a mast cell, M-A-S-T. Mast cells are also, they used to think they actually were basophils. Mast cells and basophils both release histamine and heparin, so we'll talk about the inflammatory reaction later. But mast cells and basophils especially are the ones that you'd see elevated during an inflammatory reaction. Next, we have the monocytes. So monocytes actually are a transport cell. They transport themselves, that's their payload, I guess, is themselves. They transport themselves to where they need to go. Then they become a tissue macrophage, which can leave the blood and go and do what macrophages do. Maybe you sprain your ankle. So monocytes are going to go to the ankle as part of the inflammatory reaction. They're going to turn into tissue macrophages, and then these macrophages, which basically means big eater, they're going to engulf debris and clean up the area. Or fight off infection. Let's say you have a wound, they're going to try to keep bacteria or other things from entering. All right, and the last one is lymphocytes. We'll cover them a lot more in Chapter 9. They're actually, and there's different subtypes of lymphocytes, but for now, just know they're elevated during viral infections. Excuse me. What do platelets do, and what are the other key examples or key players in the blood clotting process? Look at this real quick, make sure everything's working. Okay. So platelets, they actually stick to each other. It's called platelet adhesion. So platelets, their primary function is to get sticky. They can stick to blood vessel walls and they can stick to each other. So they form a temporary platelet plug during the blood clotting process. So it's a temporary plug. Then what happens is, so what are the other key players? That's going to be fibrinogen. So you have prothrombin becomes thrombin, which then leads to fibrinogen becoming fibrin. Well, fibrinogen is soluble in water, and fibrin is not. So what happens is fibrinogen lays over this platelet plug formed by these sticky platelets, then becomes this insoluble protein called fibrin, forming a mesh, and that's actually what's going to lead to blood clotting. So platelets play a key role early, but the actual conversion of fibrinogen to fibrin is what causes blood to clot. What vitamin is needed for proper blood clotting, and then what mineral? So most of the plasma proteins that are involved in blood clotting process, so we all have, or most of us have clotting factors, and these clotting factors are needed for the blood clotting cascade. If you don't have these clotting factors, you have a condition called hemophilia, where you would bleed very easily. But the key, so most of these proteins, these clotting factor proteins, are vitamin K dependent. So vitamin K is the crucial mineral, or vitamin, sorry, for, I don't know if I'm tired today or what, I'm going to say sorry a lot, I guess, is the primary vitamin for blood clotting. What mineral? That is calcium. Calcium plays a massive role in the blood clotting process. All right, next, what determines your blood type? So your blood type, now just so you know, there's actually dozens, probably somewhere near 50 different blood types, but we only work, for most people, unless you're severely immunocompromised or getting a bone marrow transplant or something, we're only really concerned about the ABO system and then the RH system. So if your blood type is A positive, well, it's A in the ABO system and it's positive in the RH system. So what determines your blood type is what antigens are on the cell surface. So next question, what is an antigen and what is an antibody? Antigens are cell surface markers. Every cell has them, every living cell, not just every human cell or blood cell, but antigens are cell surface markers. And the surface markers on your blood cells determine what blood type they are. Then what are antibodies? Antibodies in this context, they're going to be in the plasma, in the fluid portion of blood, and they're looking for all the other blood types. So antibodies, most of the time, they're looking for things that shouldn't be there. They're usually being produced to fight off an infection or something. But in this case, let's say you have type A blood. That means you have the A antigen on your red blood cells. It means you have the anti-B antibodies in your plasma. So if they were to accidentally give you type B blood, your anti-B antibodies would attack and destroy those blood cells. That's called the transfusion reaction. All right, so that's an antigen versus an antibody as determined by your blood type. But back to the determination, that's genetically determined. So it's a pretty neat genetic example that we will cover in more detail near the end of this course. But so A and B are both dominant genes that can be passed on from your parents. O is a recessive gene. So like if somebody has type O blood, they don't have an O antigen on their cell surface. O just basically means you don't have A or B. So if you have, so your gene, so let's say that your mom gives you an A gene and your dad gives you an O, well, you will be type A blood because A will dominate that recessive O gene. You'll have type A blood. But ABO blood system is also a neat example of codominance. If you get an A gene from your mom and a B gene from your dad, your blood type will be AB. So you have type AB blood. Both A and B can dominate O and hide it, but they can't dominate each other. So that's a neat genetic example of codominance. All right, what's the most common blood type in America? So from the ABO system, it is type O blood, and then about 84 or 85% of Americans have positive blood with the RH system. So that's going to be, so O positive would be the most common blood type in America. Who is the universal donor and the universal recipient? Let me take a drink here. So a universal donor is someone that can give blood to anybody. That would be O negative. If you have O negative blood, that means you have O in the ABO system, which means you don't have the A or B antigens. And that means that you're negative, so you don't have the RH antigen, which we'll cover in a moment. So no matter who you give that blood to, they can accept it. If they don't know your blood type, they'll give you O negative blood. Now the trick though is if you're the universal donor, you're a pretty crappy recipient. If I have O negative blood, you can't give me any other blood type than O negative. That's where the universal recipient comes in. So where the universal donor has none of these antigens on their cell surface, the universal recipient would have all of them. So A, B positive. That means no matter what blood type they give you, you're fine. They can give you A, they can give you B, they can give you O, they can give you other AB blood. But you can't give your blood to anybody else. The only people that can take blood from someone that's AB positive is someone else that's AB positive. So that's why I use the terms universal donor and universal recipient. Next, what makes the RH blood type unique compared to the ABO blood types? So the key thing here is if you have type A blood, you already have anti-B antibodies. If you have type B blood, you already have anti-A antibodies. But with RH, if you have a negative blood type, so if you're A negative, you already have anti-B antibodies, but you do not already have anti-RH antibodies. You actually have to be exposed to it once. So if I have O negative blood and they accidentally give me O positive blood, and I've never been exposed to the Rh positive blood type before or anything that looks like it, it'll be okay. But now my immune system has been sensitized. So now I will have anti-Rh antibodies. That's the key difference. So the ABO system, you're basically born with it. Now there's still theories about why that is or what actually happens. But if you have type O blood, you're going to have both anti-A and anti-B antibodies. Excuse me. But you have to be exposed or sensitized to the Rh factor, which Rh, just so you know, it used to be called D. So we had the ABO system and the D system. They call it Rh because of the rhesus monkey where they were doing the research, but it's just the positive or negative. So if you have a negative blood type and you've never been exposed to a positive blood type, you should not have anti-Rh antibodies. Which leads us to our next and last question for this chapter. When is hemolytic disease of the newborn a potential problem? So this would be the case where mom has a negative blood type. Let's say mom's O negative and the baby in her womb is O positive. Well, nothing happens during the first pregnancy. But then during labor and delivery, the placenta tears and their blood now mixes. So now mom has actually seen a positive blood type. So mom is going to develop anti-Rh antibodies, which won't cause a problem for her because she has a negative blood type. The second baby on, though, that's where you're going to have a problem. That's why they use that drug RhoGAM to desensitize the immune system. Or in this situation, during the second pregnancy, mom's immune system would attack and destroy the baby's blood. And that's why it's called hemolytic disease of the newborn. I actually had a student years ago that had this. For some reason, she couldn't take RhoGAM, couldn't get RhoGAM. It took 15 blood transfusions. 5 blood transfusions while the baby was still in her womb, and 10 after the baby was born to keep this kid alive and healthy. So this is a serious issue. RhoGAM has saved countless lives. We can't even estimate. So if you're a woman and you have a positive blood type, you never have to worry about this, because if you have O positive blood, you won't develop anti-Rh antibodies. And if you did, it would attack and destroy your blood. So it wouldn't be an issue with pregnancy. It'd be you wouldn't live. So it's always an Rh negative mom with Rh positive babies inside them, and it's the second pregnancy on where you're going to see problems. Okay, chapter 8. What is the best way to define an artery and a vein? So like when you were younger, in other classes you may have learned that arteries carry oxygenated blood or oxygen-rich blood, and veins carry deoxygenated blood. That is true in every situation but one, but there is actually, the pulmonary arteries that are going from the heart to the lungs, they're actually carrying deoxygenated or oxygen-poor blood, and the pulmonary veins are carrying oxygen-rich blood back to the heart. So because of that, the only acceptable way to define an artery and a vein is arteries carry blood away from the heart, veins carry blood towards the heart. Okay. Understand why veins and arteries look different, because they have different needs. So it's a design thing. That's what it's called, anatomy and physiology, because you learn the structure of something which determines its function. So, all right, so think about their needs. Arteries are going to have blood running through them at a very high pressure. That's why arteries, you're at much higher risk for aneurysms, because there's a lot of pressure in your arteries. That's because the heart is pumping blood through them. So arteries are going to have big, thick muscular walls, and they're going to be really elastic because their job is to put up a fight. They have to be able to withstand this pressure. So arteries are built to withstand tremendous pressure. Veins, there's barely any pressure in your veins, because blood is basically oozing or leaking back to your heart when it's being pushed away from your heart. So like, for example, the blood pressure at your aorta, it could easily be 140 millimeters of mercury, somewhere in that ballpark. The blood pressure in the inferior and superior vena cava, the two largest veins that receive blood or send blood right to the heart, the blood pressure there is going to be about 2 millimeters of mercury. So veins, they're designed to offer very little resistance. They have wide open centers or lumens. They're thinner, they're more flexible, because they can't offer too much resistance, or the blood couldn't flow through them. So that's why they look different, because they do have different needs. I couldn't think of a better word, so that's why I put needs in parentheses there. All right, or quotation marks, sorry. What happens at the capillary, and what are the different types of capillaries? So arteries carry blood away from the heart, veins carry it back. Capillaries, their job, that's where diffusion of things occurs. That's where nutrients go from the blood out into the cells. That's where waste products go from the cells to the blood, and that's where oxygen and carbon dioxide are going to be swapped. So it's the diffusion of material occurs at the capillaries. Arteries carry blood to the capillaries. Veins carry it back, but the capillaries where most of the work is done. What are the different types of capillaries? So The most common type is called a continuous capillary. They only have very small openings. But then we do have what are called fenestrated capillaries. This isn't a huge deal, but fenestra means windows. So these are going to be capillaries that have larger openings. Then we have a type of capillary called a sinusoid, which have huge gaps. So basically, continuous capillaries only let small things through. Fenestrated capillaries let small and medium-sized things through. And the sinusoids let small, medium, and large size things come through. So there are places in the body where you're going to need these different types of capillaries, but most of them are just called the continuous capillary. One more thing about that, the sinusoids, the openings are so large that every sinusoid is actually lined by immune cells. Your immune system has to keep an eye on what's going on there because large things can sneak through, and we got to make sure the things we want around. All right, what is interstitial fluid? How is it different than blood, and how is it different than lymph? So interstitial fluid is the fluid between your cells. So when you're talking about a single cell, you'll talk about intracellular fluid, the fluid inside of it, extracellular fluid, the fluid outside of it. But the fluid that's outside of your cells is actually going to be interstitial fluid in the space between your cells. How is it different than blood? Well, that kind of ties into the next thing, but it's different than blood because it does come from your blood. It's fluid that's forced out of your blood, but it's filtered out of your blood. So it's only the small things that are in blood. It's basically plasma and small proteins and nutrients and oxygen, things like that. But there shouldn't be blood cells in it. There shouldn't be large proteins in it. So it's basically blood. The small things in blood are filtered out and become interstitial fluid. And how is it different than lymph? Lymph or lymphatic fluid is actually interstitial fluid, but its name changes once it's in the lymphatic system. So they're actually identical, just named based on where they are. Which goes to the next one. So what's the relationship between capillaries and your lymphatic system? So every day, these numbers are going to be slightly different, but every day we filter out at the capillaries in our body, we filter out about 24 liters of fluid. It's pushed out by what's called hydrostatic or water pressure. So if we filter out 24 liters. And then because of osmosis, primarily because of the albumins, the protein we talked about earlier, 20.4 liters of that fluid is going to be sucked back in or reabsorbed. So we push out 24 liters, we suck 20.4 liters back in. The difference that was at 3.6 liters is going to be carried through your lymphatic system back to your heart. So it is going to return back to the circulation, but it travels through your lymphatic system first. So so why is that? Well, that's a later chapter here, but the reason for that is as lymphatic fluid travels back to your heart, it goes through your lymphatic vessels and your lymphatic organs and your lymph nodes, which are basically like checkpoints that are cleaning and filtering that fluid and also looking for pathogens. So it's a protective mechanism. All right, let's see. Why do veins need valves and arteries do not? So arteries are very high pressure, veins are not. So blood is pushed away from your heart. So blood is being pushed from my heart to my pinky toe right now. But it's got to get squeezed back in the veins. So the blood that's being carried back up from my feet is going to be moved primarily by muscle contraction. That's why if we're moving, our blood's going to be, it's not going to pool. That's why they tell you not to lock your knees when you're standing if you don't want to pass out. That's why they tell you to do foot exercises when you're on the airplane. You don't want fluid to pool. So muscle contraction is the primary way that the blood moves back to your heart. So what the valves do, or valves in any context, whether it's lymphatic vessels, veins, or in the heart, valves prevent the backflow of blood. So imagine if you were walking and your venous blood was being pumped towards your heart, but then as you stopped, it all flopped back into your ankles. That wouldn't be good. So valves, it's basically a one-way system. We can squeeze blood towards the heart, but it won't fall back as long as our valves are working. If your valves quit working, and that's where like varicose veins come from, hemorrhoids are actually varicose veins in the rectum there. So arteries don't need it because the blood is constantly being pushed. Your arteries are, your fluid's being pushed by your heart. Veins, you have to kind of milk the blood back to the heart. All right, know the key anatomical structures associated with the heart. We'll cover them as we go do the next question with the path of blood. But it's a four-chambered heart, meaning we have the two receiving chambers on the top. They're called the atria, and then we have the two large, compression chambers on the bottom called the ventricles. But we'll go through other key features. So you've got the septums. The interatrial septum is the space between the left and right atrium. The interventricular septum is the space between the left and right ventricle. When you get inside, and again, you've got videos to watch about this, but when you get inside the heart, the other key things would be like the cusps of your valves. So you've got the The tricuspid valve is on the right side of the heart between the right atrium and right ventricle. The bicuspid valve is between the left atrium and left ventricle. So the bicuspid valve is also called the mitral valve. But the cusps, how those valves work is these cusps have these cords, these fibrous or tendinous cords attached to them. Those are called chordae tendineae. I can't show you this right now, so make sure you watch the videos about the heart. And the chordae tendineae, they're attached to little papillary muscles. So those would be the key structures inside the heart. All right, know the path that blood takes as it flows through the heart. There's a video called The Path of Blood Through the Heart. Make sure you watch that one. But I'll walk you through it. We'll start at the superior and inferior vena cava. So that's going to be the veins that dump blood into the heart. So they're going to drain into the right atrium. Then when the right atrium contracts, blood's going to go through the tricuspid valve to the right ventricle. Then when the right ventricle contracts, it's going to push blood through the pulmonary semilunar valve to the pulmonary trunk, and then it's going to go to the pulmonary arteries, and it's going to go to the lungs and drop off CO2 and pick up oxygen. Then it's going to come back through the pulmonary veins, and it's going to dump into the left atrium. When the left atrium contracts, blood's going to be pumped through the bicuspid valve, also known as the mitral valve. Now, if you want to know why that is, a mitre is a bishop's hat. So somebody thought that the mitral valve looked an awful lot like a hat that a bishop in the Catholic Church would wear. So from the left atrium through the mitral valve or bicuspid valve to the left ventricle. Then when the left ventricle contracts, blood's going to go through the aortic semilunar valve to the aorta, and then it's going to go everywhere else. And it's going to work its way through your body wherever it's heading. It's going to come back. It's going to end back up at the inferior and superior vena cava, or one or the other. And it's going to be back at the right atrium. So that's the path blood takes through the heart. So that's very visual, so make sure you watch that video. All right. Why is the left ventricle so much thicker than the right ventricle? It's because it has to pump blood greater distance with greater pressure. The right ventricle has an easy job. It has to pump blood to the lungs and back. They're sitting right next to each other, like literally touching each other. So the left ventricle has to pump blood from the top of the head to the bottom of your feet. So that's why the left ventricle is going to be about 300% thicker. It's a bigger, stronger muscle because it has to work harder. If you curl 2-pound weights every day, your arm's going to be pretty small. If you curl 30-pound weights every day, your arm's going to be big. It's the same with the heart. The left side of the heart has to work that much harder. All right, know the key steps of the cardiac cycle. Really what I want you to understand here is that we have atrial systole. So systole, it kind of leads into the next question. Systole means contraction, diastole means relaxation. So you're going to have atrial systole. That's when the two atria are contracting. You're going to have atrial diastole. That's when the two atria are relaxing. Probably the most important step is atrial or ventricular systole, when the two ventricles are contracting. And then we have ventricular diastole, where they're relaxing. So the cardiac cycle is a combination of systole, which means contraction, and diastole, which means relaxation. So why are they clinically significant? Well, you've heard your blood pressure. Your blood pressure is supposed to be 120/80. That is your systolic versus your diastolic blood pressure. So the 120 is when the heart is beating. The 80 is when it's resting. But the other reason it's significant is you have a high heart rate. So if your pulse rate is in the 90s or higher, the concern there is the faster your heart has to beat, the shorter the diastole. So if you have high blood pressure, which you may have no symptoms at all, but if you have high blood pressure, it's putting your heart never gets a rest. There's always pressure on it. It doesn't get to fully relax. And if you have a high heart rate, the rest phase of the cardiac cycle is shortened. So you can see why having high blood pressure and a high pulse rate are as a recipe for disaster. All right. What actually makes the heart sounds that you hear when auscultating the heart? So I'm not going to worry about, we'll just call them lub-dub or lub-dup. They're actually called S1 and S2, and then you've got the S3, S4, which you can hear as well. But generally, when you hear the heart, it is the closure of the valves that you're hearing. That's what I want you to know. There's certainly some more detail there, but it's actually when the valves snap shut is what you're hearing with the lub-dup, lub-dup. It's usually lub-dup, D-U-P-P, lub-dup, lub-dup. So when you're hearing that, when you're auscultating, which means listening to the heart with a stethoscope, that's what you're hearing, is the closure of the valves. All right, know the steps of the conducting system of the heart. Hey. How are you? Good. All right, sorry about that. Okay, so know the steps of the conducting system of the heart. That's the electrical portion of the heart. So that starts with the SA node, which is the sino or sinoatrial node. That's in the wall, the back wall of the right atrium. So it goes from the SA node to the AV node, which stands for atrioventricular. So the SA node is your pacemaker. It sets the pace of your heart. The AV node is between the two atria on top and the ventricles on the bottom. So basically, you send an electrical current through both atria at the same time. Now at the AV node, there's a 100 millisecond delay. And what that does is gives the two atria time to contract before the ventricles do. Because the atria, their job is to fill the ventricles. So that 100 millisecond delay is very important. Then it goes from SA node to AV node, then the AV node to the AV bundle to what are called the bundle branches. They were called the branches of HIS, H-I-S, when I was a student. Then they come back up to the Purkinje fibers. So know those steps. There's, of course, a video on that. But that's how it goes. So basically, your two atria are going to depolarize and then contract. There's a 100 millisecond delay to give them time to do that. Then your ventricles will depolarize and kind of squeeze and contract from the bottom, kind of like wringing out a sponge. All right, which relates then, what can you see on an EKG or an ECG? It is actually now an electrocardiogram, ECG, but EKG because it was a man named Einthoven in Germany that invented the technique, and it would be a K there. So when you say EKG, you're really kind of an homage to this Einthoven guy. But it measures the electrical activity of the heart. So it measures that conducting system, and you can learn a lot from an EKG. You can tell like where the heart is sitting, how big it is there evidence of occluded blood vessels. Like a good cardiologist, well-trained cardiologist can learn a lot from an EKG. But big picture is it tells you it measures the electrical activity of the heart. Now the actual parts of an EKG, you've got the P wave, the QRS complex, and the T wave. So the P wave is measuring when the atria depolarize, so when it depolarizes electrically. So when the SA node sends that signal towards the AV node. Then the QRS complex is when the ventricles are going to depolarize. So now the ventricles are depolarizing. And the T wave on an EKG is when the ventricles repolarize. So notice what you didn't see there was when the atria repolarize. And that's just because it's hidden by the QRS complex. So no big deal. All right, lastly, define cardiac output. So your cardiac output, is how much blood you move per minute. So it is your stroke volume, which is how much blood you move per contraction, per beat, times your heart rate. How many times is your heart beat in a minute? So that is your cardiac output, which is normally 5 to 6 liters a minute for someone sitting there at rest. Okay, next chapter. What type of microbe are antibiotics effective against? Antibiotics kill bacteria. And they're not even doing that super well anymore. I mean, they still do. But that's very important because if you're being given an antibiotic for a viral infection, that's not how they work. So I just wanted you to know that. Are viruses alive? Viruses are clearly very important. We have less treatments to kill viruses. I use the word kill, I shouldn't, but than we do bacteria. But they're not alive. They're parasites. They don't have their own metabolism. They don't all have DNA. And they don't have a cell. They're not made of cells. So for many reasons, they're not alive, but it doesn't mean they're any less significant. We just can't technically say they're alive. So we shouldn't technically say you kill a virus, you inactivate a virus. All right, what is lymph? How similar is it to blood or other body fluids? So remember, lymph is interstitial fluid. It's just once it enters the lymphatic vessels, it's now called lymph. So it is blood. That's only the small stuff that's in blood. So lymph is very similar to blood plasma. It's like the fluid portion of blood with all the larger things like the plasma proteins removed. So it's basically that's what makes it similar to blood and other body fluids. What makes your lymphatic system different than your circulatory system? So two things. Number one, it doesn't have a pump. Your heart pumps blood through your circulatory system, at least through your arteries. Your lymphatic system doesn't have that. So your lymphatic vessels look like veins. They have valves, and muscle contraction, muscle compression is what moves the fluid. So there's no heart. Number two is it's not a circle. So your circulatory system moves blood away and then towards. Your lymphatic system, all the vessels run in one direction. They're all one-way roads. They all carry things from the outside in. So those are the two reasons they're different. What is unique about the thymus gland compared to most organs? The thymus gland is very important when you're young and becomes less important. So it actually shrivels up and atrophies and becomes fibrotic. So let's say when you were born, your thymus gland might have been like 1.5 ounces. By the time you're 50, it might be like 0.4 or 0.3 ounces. So that's kind of a unique thing. Now, what's it do? The thymus gland is where your T cells mature. So they're called T cells for a reason. T cell means thymus-dependent cell, whereas B cells are bone marrow dependent. All right, what are the 4 cardinal signs of inflammation? So these are going to be caused by those inflammatory chemicals we already mentioned, histamine and heparin, but it's redness, swelling, heat, and pain. So pain is a sign of tissue damage, whether it's an infection, trauma, doesn't matter. Redness, swelling, and heat all come from the fact that there's more blood in the area. It's red because more red blood. It's swollen, there's more fluid there because this extra blood is coming to the area. And it's hot because your blood is 100.4 degrees. It's warmer than the rest of your body. So redness, swelling, and heat are all just signs of increased blood flow. But the 4 cardinal signs of inflammation are redness, swelling, heat, and pain. I won't ask you to know them in Latin like I had to, but just know that. All right, what is the difference between antibody-mediated or humoral immunity and cell-mediated immunity? So real quickly, it is B cells that power antibody-mediated immunity, and it is T cells that power cell-mediated. The key difference, cell-mediated, think hand-to-hand combat. It's a T cell attacking a cell directly. Antibody-mediated or humoral immunity, it's your body fluids, humors or fluids. So it's the B cells producing antibodies that go and attack other cells. So think cell-mediated immunity, think direct contact, direct attack. Antibody-mediated, think indirect attack. Here's how I think of it. I don't know if you ever played the game Clash of Clans or ever watch shows about knights and castles and all that business. A T cell, cell-mediated immunity, that'd be a soldier with a sword, like a knight with a sword, directly attacking something, a pathogen. Antibody-mediated or humoral immunity, think archers. They're firing off antibodies that are attacking other cells. They're not directly attacking them. All right, what does a B cell become before it starts making antibodies? So this is, it's very important. B cells, when they activate, they actually become plasma cells, and those are the cells that make antibodies. Know the key features of the 5 classes of antibodies. So you've got, how you remember them is gamed, IgG, IgG, IgA, IgM, IgE, and IgD. Okay, so we'll go in that order. IgGs, this is going to be like 70 to 75% of the immunoglobulins, so make sure you know they're the most common for sure. The other big thing about these, all 5 types of antibodies can be transferred in breast milk, but IgGs are the only ones that pass to the fetus. So clinical example, that would be the anti-Rh antibodies that would cause that hemolytic disease of the newborn. Those would be IgG antibodies. IgAs, these are every glandular secretion, every part of your body lined with the mucous membrane is going to have these. So they're actually called secretory IgA antibodies. So we're talking tears, saliva, the mucus in your gut, all glandular secretions, even semen and stuff, they're going to have these IgA antibodies. All right, so that's IgG and IgA. IgM, these are the largest ones. They're called pentamers. Those structurally they're the largest, and they show up first in the primary immune response. That's kind of important. But they're your ABO blood types would be IgMs. That's just kind of the example there. So they're structurally the largest. They're not super effective, but they do show up first in the primary immune response. So that's IgG, A and M. IgE and IgD are both pretty rare. IgEs, think inflammatory response. So they play a role when histamine is released. It leads to these IgE antibodies that are actually being stimulated. So it's B cells actually have these IgE antibodies on their surface, and that's what's going to lead to like mast cells and basophils and some B cells releasing antibodies. So they're actually on the cell surface. The last one is IgD. I don't even know, they're on the surface of B cells as well. They probably play a role in activating B cells, but it's not clearly understood, so they're not a super important one. So that's IgG, most common, and the only one that crosses the placenta. IgA, found in glandular secretions. IgM, structurally the largest, and the first to show up during the primary immune response, which means the first time you're exposed to something. IgEs, think inflammatory response. They're actually on basophils, and that's what triggers. So like an antihistamine would actually stop these IgE antibodies, stop the basophils if they're stimulated from releasing histamine. And then IgD, think on the surface of B cells and most likely play a role in activating them. Okay, what does each type of T cell do? Helper T cells, cytotoxic T cells, and memory T cells are the key ones here. So helper T cells, I call them the generals because they don't attack things directly, but they activate your immune system. Helper T cells, they actually activate other T cells and B cells. So both parts of your immune system, both the antibody-mediated and cellular-mediated immunity, helper T cells are required. That's why HIV destroys your immune system. It attacks the helper T cells, the only cell type that it really does affect. Cytotoxic T cells, those are your soldiers. They're the ones actually directly attacking pathogens. Memory T cells, whenever you see the word memory in response to your immune system, that's how your immune system remembers things. That's how you develop immunity. If you didn't have memory cells, we would never have immunity. Every time we're exposed to measles or the flu or anything, we would get it. We wouldn't develop that memory. So those are the key ones. There is another type. They're called regulatory T cells. It used to be called suppressor T cells. They play a role in making sure the immune system doesn't overreact. And they're very important when it comes to allergies and autoimmune diseases, but you don't need to know them for this course. All right, what's the primary function of the natural killer cell? That's a great name for a cell, by the way. But natural killer cells, they're involved in what's called immunological surveillance. They can attack and destroy virus-infected cells, but most importantly, they can attack and destroy cancer cells. So that's going to be their primary function. When you think cancer, definitely think natural killer cells. All right, why do we often get sick the first time we are exposed to something, but not the second? What role can vaccines play in this? So that's this whole idea of primary immune response versus secondary immune response. So the first time you're exposed to a pathogen you've never seen before, it takes, the IgM antibodies will show up in a few days, but it takes 10 to 14 days to mount a complete immune response, which means that organism's going to have made you sick in most cases. But the second time you're exposed to something, the IgG antibodies show up right away and much more powerfully. So that is how we develop immunity, because of this memory. So that's why we get sick the first time we're exposed to something, but not the second. So what role can vaccines play in this? Well, a vaccine can actually show your immune system parts of a pathogen. This is how I define vaccines to people that don't know science. The first time you've been exposed to an actual pathogen in the world is the second time your immune system has seen it, because we've shown your immune system what to look for, basically created a wanted poster for that organism. So that's how vaccines work. They bypass the need for that primary immune response, or at least in the real world. So you might be thinking here, like, wait a minute, I get the cold every year, but that's because there are reasons that we get the same diseases over and over. Number one, there are potentially thousands of viruses that cause the cold. And number two, they mutate and change so quickly that even if you got the flu last year, the flu next year is going to look different. So this is not a perfect system, but that is how it works. All right, what is active immunity versus passive immunity? So active is something your immune system has to actually do. Passive is something that's kind of done to you. So I'll quickly go through them here. We have naturally acquired active immunity. That is, you get exposed to a pathogen. You get sick or you don't. Maybe you didn't get exposed enough to get sick. You get sick, and if you don't die, you're now immune. That's naturally acquired active immunity. Naturally acquired passive immunity would be breast milk. So your mom was passing on her antibodies, her immune system, through breast milk. But that's naturally acquired. It happened in nature prior to technology, and it does help your immune system. So passive immunity is when you actually are given antibodies. So think of it that way. Active immunity, you're exposed to antigens and your body responds to them. Passive immunity, your body is given antibodies directly. So then we have artificially acquired active immunity. That's going to be vaccines. The vaccines have the antigens that your immune system will then develop antibodies and immune cells against, and memory too. Artificially acquired passive immunity would be antibody therapies. So it'd be like if you get Ebola, they would give you Ebola antibodies to actually try to keep you alive. You get bit by a snake, they're not going to say, you know what, if you survive this snake bite, 14 days from now you'll have antibodies. They'll actually give you the antibodies. So sometimes they actually do both. Let's say you step on a rusty nail, they can give you a tetanus shot, the vaccine, just to keep your booster updated, but they'll also give you the tetanus immunoglobulin. They'll give you an immunoglobulin or an antibody therapy, a form of passive immunity as well. All right, know the key features of HIV. What kind of virus is it? What cells does it attack? Should be, well, I guess that's okay sentence-wise. What are the phases of HIV infection? So what kind of virus? It's a retrovirus. You got a few videos, 2 or 3 videos you should be watching about HIV. It's a retrovirus. That's because it's an RNA virus that can actually turn itself into DNA. Makes them very hard to kill. Hepatitis C is another key example there. What cells does it attack? It attacks the CD4 positive T cell, which is the helper T cell. So that's the receptor the HIV virus is looking for. That's the lock the HIV virus, is the key for. And then what are the phases of HIV infection? There's 3 phases. Phase 1, either no symptoms or chronic lymphadenopathy, which would be chronic swollen lymph nodes. Phase 2 is when you start to show signs of having a compromised immune system as your T cell population drops. Phase 3 is AIDS. So once your helper T cell count drops below 200 per microliter of blood, you now have AIDS. So HIV is the human immunodeficiency virus. AIDS is the acquired immunodeficiency syndrome. So it's acquired because there is actually genetic versions of the disease. If you've ever seen the movie The Boy in the Plastic Bubble, now I'm assuming many of you were younger than me. John Travolta was like 9 years old in it, so that's how old it is. But so there are genetic conditions that can look a lot like AIDS. This is an acquired immunodeficiency. All right. Next 2 chapters, not quite as much stuff, or I'm sorry, next 1, not as much stuff, and then chapter 14 is a big one. Why do we need to breathe? Excellent question. We need to breathe because we have to get oxygen, and that's because our metabolism requires it. If you understand, the electron transport chain, how we turn food into fuel, it is glucose or whatever fuel source you're using plus oxygen. Now, if you want to know the details of why, oxygen is called your final electron acceptor, but don't worry about that. We also need to, so that's why we breathe in. Why do we breathe out? We have to get rid of carbon dioxide. CO2 is a weak acid, so if we keep too much carbon dioxide in our body, our blood pH will drop and we will develop a condition called acidosis, which can be fatal. So we have to breathe in because we have to get oxygen for our metabolism. We have to breathe out because carbon dioxide is a waste product that if we don't remove it, can kill us. All right, what separates the upper and lower respiratory tracts? That's the larynx. Everything above the larynx, so your nasal cavity and the pharynx, and your sinuses and everything, that is your upper respiratory tract. From the larynx down, so the larynx down to the trachea, to your bronchial tree, all the way to the ends, the gas exchange surface of your lungs, is the lower respiratory tract. So why do these terms matter? Well, the upper respiratory tract is more common to have infections and problems with it, sinus infections, etc. The lower respiratory tract is much more dangerous. So we're talking pneumonia, bronchitis, things that can be fatal. All right, what is the main function of the nasal cavity in regards to breathing? So when you breathe through your nose, the point is to create turbulence, and air is going to swirl around inside your nose, inside your nasal cavity. And as the air swirls around, here's the 3 key words: filtered, warmed, and humidified. So you're preparing air for your lungs by filtering it, warming it up, and humidifying it. What is the opening to the trachea called? What protects it when you swallow? So the opening to the trachea, which the larynx is built around, is called the glottis. So the glottis is the opening to the trachea. What protects it? A flap of elastic tissue called the epiglottis. So when you swallow, which we'll cover in the next chapter, the epiglottis flips down over the glottis of the trachea, which covers it so that when you eat or drink, food goes down your esophagus, not into your trachea, down the wrong pipe, as you'd say. All right, what is the primary gas exchange surface of the lung? So everything I just mentioned, the larynx, trachea, primary bronchi, secondary bronchi, the whole thing, that bronchial tree, that's all designed, it's called the conducting system. It's designed to move air. But where are the gases exchanged? Where do we trade oxygen for carbon dioxide? That's called the alveoli, A-L-V-E-O-L-I, the alveoli of the lungs. All right, what is the primary muscle of respiration or breathing? That is the diaphragm. So the diaphragm is the muscle that actually is the border that separates your thoracic cavity, which is where your heart and lungs are, from your abdominal pelvic cavity. So everything below the diaphragm is in the abdominal pelvic cavity. But 75% of a normal quiet breath in is actually caused by the diaphragm contracting. So the diaphragm is the primary muscle we use to breathe in. I won't go into detail here, but interestingly, we don't use any muscles to breathe out. Now, if you're forcing air out because you're exercising or whatever, or you have a medical condition, then you're going to be using lots of muscles. But a normal quiet breath in just involves the diaphragm and then your external intercostals, the muscles between your ribs. And a normal quiet breath out requires no muscles. Just those muscles relax and the elastic tissue basically snaps shut, kind of like if you take the air out of a balloon. All right, what is tidal volume? What is vital capacity? So there's a video you guys should be watching about your lung volumes and capacities, but tidal volume is how much air you move during a normal quiet breath, and it's usually going to be about 500 milliliters. So a normal breath in, normal breath out, that's your tidal volume. Vital capacity is how much air you can move. If I give you a spirometer or a peak flow meter and I say take the biggest breath you can and blow as hard as you can, blow all the air out you can, that's your vital capacity. It's not your total lung capacity because you have, there's lung tissue that you can't, there's air in your lungs you can't control. That's going to be called your residual volume. And even if both your lungs deflated, you'd have a tiny bit of air left. That's called your minimal volume. So I want you to make sure you're clear on that. The vital capacity is not the same thing as total lung capacity. Your lungs are bigger than what you can control. The vital capacity is how much you can control. So right now, take the biggest breath you can and you blow it out as hard as you can. You're bent over, red in the face. That's your vital capacity. But there's still air in your lungs. That's the residual volume. Okay. What is the key way oxygen is carried in your blood, and how about carbon dioxide? So almost 100%, 98% or so of oxygen is carried on hemoglobin. CO2 is different. So CO2 can be carried in three ways. It can be dissolved right in your plasma. It can be carried on hemoglobin. So we know hemoglobin carries oxygen, but it also can become carbaminohemoglobin and pick up carbon dioxide. And it can just be carried in your red blood cells, or it can be carried as carbonic acid. So there's actually three ways you carry carbon dioxide back to the lungs, only one real way you carry oxygen towards your cells. Last one for this chapter, is your brain more concerned about oxygen levels or CO2 levels? And the answer is CO2. Your brain is, when it comes to breathing, your brain cares most about your pH, how many hydrogen ions there are. And remember, carbon dioxide is an acid, so we have to control how much carbon dioxide we get rid of more than how much oxygen we need. Look at like a Navy SEAL can hold their breath for minutes. We have tons of oxygen. Every red blood cell can carry like 1.2 billion molecules of oxygen. We have extra oxygen in our blood. You should anyways, unless you have anemia. But we've got to get rid of CO2. So your breathing rate is primarily controlled by your carbon dioxide levels and your pH, not oxygen levels. All right, skipping chapter 14 and 15. So 15 will be kind of short. I know this is getting long, but this is the only time I get to lecture to you guys. So I'm trying to make it count. All right, chapter 14, what are the three key functions of the digestive system? Hint, the first is digestion. So it's digestion, absorption, elimination. Digestion is the chemical breakdown of whatever you eat into the smallest possible units. Absorption, carrying it across your GI tract, primarily in your small intestine, into your blood or your lymphatic fluid. Elimination is getting rid of what you can't digest and absorb, which should just be fiber. When you defecate or poop, it should be fiber and dead bacteria, basically. All right, digestion, absorption, elimination. Know the key features of each part of the GI tract, like 10,000 foot view. This will all be in your digestive system overview. So you got the mouth. The mouth mechanically breaks down food, and then mixes it with saliva, which lubricates it and starts the chemical digestion process. The pharynx, the throat, and your esophagus, they're just hollow tubes that carry the food to the stomach. The stomach's job is to use stomach acid and protein digestion enzymes to start to digest and unravel proteins, primarily. That's why the protein content of a meal determines how long it sits in your gut. Then you have your small intestine. Small intestine is where almost all the digestion and absorption takes place. Large intestine, that's going to be where we reabsorb water. We do absorb a few other things, but the key things are we reabsorb water, and that's where we temporarily store feces until we defecate. So that's like 10,000 foot view. What are the different names for food as it passes through your body? So it's food when you put it in your mouth. Once you've chewed it up and turned it into this oval lubricated mass, it's now called a bolus, B-O-L-U-S. Once it's become this like syrupy liquid after the stomach's done with it, it's called chyme, C-H-Y-M-E. And then once it's in your large intestine and it's had the water taken out of it and you're ready to defecate it, it is called feces. All right, what are the accessory organs of the digestion? That's going to be salivary glands, liver, gallbladder, pancreas. Compare peristalsis and segmentation. So they both involve the smooth muscles of your gut. Segmentation is a churning. So if you're churning and mixing food like in the stomach or the duodenum, the first part of the small intestine, that's called segmentation. Peristalsis, think muscle contractions that move materials from one place to another. So like swallowing involves the peristaltic waves of contractions that carry food from the back of your throat to your stomach through the esophagus. So that's peristalsis versus segmentation. So why is saliva important? Well, there's lots of reasons. It's primarily water, but so it lubricates your food, so it makes it easier to swallow. It has antibodies in it. It has lysozymes in it that can kill bacteria, so it has immune functions. But it also has digestive enzymes. It has salivary amylase, which starts the breakdown of carbohydrates. If you want to try it in a little experiment, take something like a saltine cracker and chew it until it starts to taste sweet. What's actually happening is the salivary amylase is starting to break down the large carbohydrates in the saltine and making them shorter so they become sweet. And then we have lingual lipase that starts the digestion of fat. So saliva has tons of functions. All right. What are the key steps in the swallowing process? Here's how I like to look at this. There's 3 places food or drink can go. Up out your nose, if you've probably had that happen to you before. Down the wrong pipe into your trachea. Or down the esophagus where it's supposed to go. So swallowing is all about closing the other 2 exits. So when you swallow, your food is pushed up to the top of your roof of your mouth and your tongue basically pulls it back. That flips the soft palate, including the uvula, your grape-like barf button I like to call it. That covers the nasal cavity. So food or drink shouldn't get out your nose. As you swallow, your throat climbs up to basically meet that food. As that happens, the epiglottis flips down and covers the glottis, closing your trachea. So swallowing is all about closing the other 2 potential exits or entrances, and then the food's going to travel from your pharynx down your esophagus into your stomach. So that's the key there. All right, what are the 3 parts of the small intestine? Do they have the same function? So I'd say no. First, we have the duodenum. Some call it the duodenum. I call it the mixing bowl. That's going to be where this syrupy chyme from the stomach is going to mix with bicarbonate from the pancreas, which is a buffer to help get rid of the stomach acid, digestive enzymes from the pancreas, and then bile from the liver and gallbladder. So they're all going to get churned together in the duodenum. So think mixing bowl. The jejunum, the second part of the small intestine, this is going to be where like 80 to 90% of digestion and absorption take place. So that's the key from a digestion and absorption standpoint. The ileum is honestly just kind of famous for being the longest, but the ileum would be where like B12 is absorbed, where bile is reabsorbed, these kind of things. So it has some functions, but it's pretty minor. So those are the 3 parts and kind of their main functions. All right, what percent of the pancreas produces insulin? I ask that because when you think of the pancreas, you think of insulin and diabetes, but only 1% of the pancreas is endocrine. 99% of it is an exocrine organ, a digestive organ. And so that leads to the next question. What role does the pancreas play in digestion? It produces bicarbonate, the buffer I just mentioned. Your stomach can tolerate stomach acid, your small intestine cannot. So the bicarbonate will buffer those acids, and it produces most of your important digestive enzymes. So it's primarily a digestive organ. If your pancreas doesn't work, my wife works in digestive oncology primarily, but she sees patients that have severe pancreatic insufficiency. This is not a joke and not to be crass, but if your pancreas doesn't work, if you eat a salad, you will poop out leaves. You'll poop out lettuce leaves. Your food will be almost completely undigested when it passes through you. So how they treat that is they use high doses of digestive enzymes. All right, what organ receives the nutrient-rich blood from the majority of the GI tract? So once you've digested your food, now you absorb it. Most of it, everything but fat. Fat goes through your lymphatic system and then works its way to the blood, but everything else is going to go right to the liver. Your liver is receiving almost all of the blood from your GI tract. So it calls dibs. It calls dibs on proteins, vitamins, minerals, carbs. The liver is going to work with those structures and store some and change some and get rid of others. So it calls dibs on everything but fat. Now that's because your liver doesn't care about fat. Your liver doesn't. Your liver can turn carbs into fat, but doesn't need to or like to. So your fat cells sadly get dibs on fat. When you eat fat, it goes through your body first. Your fat cell says, "Hey, I'll take some of that fat," and then it works its way to your liver later. So the answer there is liver. Does bile digest fat? No. Your liver produces bile. It's stored and concentrated in the gallbladder, but it doesn't digest fat. It emulsifies fat. Emulsification is, oil and water don't mix. So you take these large droplets of fat that aren't mixing with water, and you break them up or emulsify them into thousands of tiny ones. So its job is to increase the surface area of fat so that it can be digested. So it's kind of a trick question, but bile does not digest fat, but it makes it way more digestible. All right, what's the difference between, now kind of some nutrition stuff, what's the difference between a saturated and unsaturated fat? So those terms actually, it's because saturated fats are saturated with hydrogens. What that means is there's no double bonds, and these are going to be solid at room temperature. They're very stable fats. Unsaturated fats are going to have one or more double bonds. So a monounsaturated fat, like what you'd find in olive oil, has one double bond. Polyunsaturated fats, like you find in fast food and vegetable oils and stuff, they're going to have more than one. So that's going to be the difference, saturated versus unsaturated. So there's obviously a huge debate about this, but there are unsaturated fats that are really bad for you, and there are some that are really good for you. There are saturated fats that are bad for you and good for you. It's tricky, very, very nuanced. That's why hopefully you'll take a nutrition course too. What is the difference between a sugar and a complex carbohydrate? That's going to be size. So your monosaccharides, your sugar units made of one unit, or your disaccharides made of two. So this would be like glucose and sucrose, which sucrose is table sugar. Those are simple sugars. Your polysaccharides, made of long chains of sugar units, are going to be complex carbs. They're going to be starch, which is like how plants store energy. Glycogen, how animals store energy, including us. We store glycogen. Glycogen is how we store glucose in our liver and our muscles. And the third one is cellulose or fiber. Those are your complex carbs. What's the difference between a complete and incomplete protein? So if you eat a complete protein, which is going to be primarily, there are other exceptions, but primarily all animal products are going to be complete proteins, and soy and quinoa, those are complete proteins. That means they have all the essential amino acids, all the amino acids we have to eat because our body can't make them. Incomplete proteins do not. So like if you're going to be a vegan and you're not going to eat animal products and you're not going to eat soy or quinoa, you have to eat what are called complementary food combinations. Like if you're going to eat legumes, beans, you have to eat rice with it. Because rice is missing amino acids, beans are missing amino acids, but they're missing different amino acids. They actually can make up for each other. So that's called a complementary food combination. It's so much easier now to be a vegan because you can just take pea protein powders and those kind of things. But what's the key difference between vitamins and minerals? So they're both very important. They both power our enzymes, our metabolic processes. But vitamins are organic and carbon-based, and minerals are inorganic. So minerals you're going to see on the periodic table. Vitamins are carbon-based structures that are built. The other key difference, you can make, living things make vitamins. They don't make minerals. Minerals are found in the soil. So, if you eat a healthy plant or animal, it can have been making vitamins for you. But if the minerals aren't in the soil, they're not going to be there. Let me give you an example, like Brazil nuts. Brazil nuts are famous for how much selenium they have, but only if they're actually grown in Brazil, because the soil in Brazil is really high. Brazil nuts don't make selenium. They extract it from the soil. So that's another key difference. What's the difference between a water-soluble vitamin and a fat-soluble vitamin? So if you look at in your chapter 14 resources, I gave you a list of vitamins and minerals I have my nutrition students know, so you don't have to know any of that detail, but just in case you wanted it. Except for one exception, water-soluble vitamins cannot be stored. Any excess is excreted in your urine. Fat-soluble vitamins can be stored. So that's the key difference. The exception there is B12. Your liver should be able to store up to 5 years of B12, even though it's a water-soluble vitamin. The other thing with fat-soluble vitamins, though, is you have to eat them with fat. This is not just fat-soluble vitamins either. Things like carotenoids too. If you have like beta-carotene or vitamin A without any fat, you're going to absorb it hundreds of times, several times less, hundreds of % less than if you had fat with that meal. So you have to have fat with a meal to absorb the fat-soluble vitamins. Water-soluble vitamins, that's not the case. All right, what's the difference between a major mineral and a trace mineral? It's not about importance. Let's see where we're at here. Okay, so we're at an hour. We're getting there. It has to do with how much you need. If you need more than 100 milligrams a day, it's a major mineral. If you need less, it's a trace mineral. But it has nothing to do with how important they are. Some of the most important minerals, you need microgram quantities, very small amounts. So, okay, last chapter, and it's a short one. Why do we need a urinary system, and what is urine? So I'll start with that. Urine is filtered blood. You don't think of it that way, but that's why urinalysis is such an important diagnostic test, because it tells you about things that are going on in the blood. So urine is filtered blood. So why do we need a urinary system? Your metabolism is trying to kill you. Every second of every day, your metabolism is churning out waste products that have to be dealt with. And urinary system is how we get rid of most of them. The big ones would be like urea, which is the leftover from whenever we use protein to do things. We have to deal with the nitrogen. Creatinine, that's just when your muscles contract, they release creatinine. Uric acid could be something. If uric acid levels build up too much, you can get gout. These are the kind of things that we have to deal with. All right, how much? This is a trick question kind of, but how much water do you need to drink every day? I'm not asking for 8 glasses a day, anything like that. So make sure you watch the video about balancing water gains and losses. So how much water do you have to gain every day? Well, you tell me how much you lose every day. If you're a roofer working in a hot, humid environment and you exercise 2 hours a day, you need to drink a lot more water because you're losing a lot more water. So real quickly, we gain water by drinking it, eating it, and making it. Your metabolism, actually, the equation for your cellular respiration is glucose plus oxygen makes carbon dioxide, water, and ATP. So you actually make some. So we have to balance that with losses. And we lose water through urination, or micturition it's called, sweating, evaporation through our breath, and then some from feces. So how much water you're losing should determine how much you're making. That's the kind of trick. Now, if you want to know my advice on how much water you should be drinking, ask your body. So if you're urinating every 3 hours, give or take, and your urine is pale yellow, like a pale straw color, then you're hydrated. It doesn't matter. Like for one person, it might be 5 glasses a day. For someone else, it might be 12. So everyone's going to be different. We know we're all different. So that's how I like to look at it. All right, know the function of each portion of the nephron. So the glomerulus, that's going to be the knot of capillaries that's sitting in the glomerular capsule. That's where fluid is going to be filtered out. 50 gallons a day of fluid is going to go from the capillary beds, this knot of capillary beds called the glomerulus, into the glomerular capsule, which was called the Bowman's capsule when I was a student. So the glomerular capsule's job is to catch that. So those two things together form something called the renal corpuscle, and their job is filtration. So they filter all this fluid material out of your blood, 50 gallons a day. Then we have the actual proximal tubule, which I call the proximal convoluted tubule. Its job is reabsorption. It's going to reabsorb 70% of that water. It should absorb 100% or almost 100% of the vitamins, minerals, glucose, that kind of stuff. So its job is to reabsorb stuff we don't want to get rid of. The problem with filtration at the corpuscle, the glomerulus plus the capsule, is it filters out things that are small and bad, which is great, but also filters out things that are small and good, which is terrible. So the proximal convoluted tubule, or proximal tubule, its job is reabsorption. So in a word, the glomerulus and capsule, that's filtration. The proximal tubule, think reabsorption. It pulls back all that good stuff. The nephron loop, which is what it's called now, but I noticed the book calls it the loop of Henle, which is fine. That's what it used to be called. But the nephron loop or loop of Henle, think concentration of urine. It's going to reabsorb a bunch more water. Now, not all of it. By the time we're through the nephron loop and the distal tubule, we still have like 27 liters of urine a day, which we can't get rid of that much. So think concentration of urine is the nephron loop or loop of Henle's job. Distal tubule, in a word, is secretion. And the collecting ducts, their jobs, they take all the fluid from the individual nephrons. Their job is to determine the final concentration of urine, which leads into the next question. And that's going to be what the collecting ducts, they're going to drip into the tubules inside your kidneys, which is going to lead to your ureters, lead to the bladder, and then the urethra. All right, so what role does antidiuretic hormone play in urine production, and what about aldosterone? So if it weren't for antidiuretic hormone, if you didn't have it, you'd have a condition called diabetes insipidus, and you could make up to 27 liters of urine a day. A liter of urine is like the fullest bladder you've ever had, so imagine having one of them an hour or more than that. So ADH determines how much extra water you have, or if you have any extra water. And your ADH levels, which stands for antidiuretic hormone, that's going to determine how much water you reabsorb. Antidiuretic hormone opens water channels, so you reabsorb more or less water. So the higher your ADH levels, the lower the urine production. So that's how your body conserves water. Aldosterone works with ADH. We haven't covered the hormones yet, but aldosterone causes the reabsorption of sodium, and then water follows it through the process of osmosis. So those two hormones, if there's a lot of it, you're gonna reabsorb more water and make less urine. If there's not a lot of them, you're gonna make more urine. So if you're really hydrated, your body will say, just let some of the water through, that's fine. And these hormones won't be as active. One more significant thing, just clinically relevant with aldosterone, it appears insulin plays a major role in aldosterone levels. This is why if you go on a crash diet or a low-carb diet, you're going to lose several pounds the first week because your aldosterone levels are low, so you're going to lose more water. You're reabsorbing less sodium, which hence reabsorbing less water. So that's why you lose a lot of water weight when you first start a diet. All right. What role do your kidneys play in the maintenance of blood pressure? So your kidneys care about blood pressure greatly. they're very sensitive to drops in blood pressure. If your kidneys function, or how you measure kidney function, is something called the glomerular filtration rate, how much it's filtering, but filtration is pressure based. So a 20% drop in blood pressure to your kidneys could shut off your kidneys. So if your kidneys notice a drop in blood pressure, they're going to send out signals to raise blood pressure. The biggest one is called the renin-angiotensin system, which is going to lead to more ADH, more aldosterone. You're going to be thirsty. I won't go through all that. But your kidneys play a major role in maintaining blood pressure because they need it. If your blood pressure at your kidneys is dropping, they'll also stimulate another hormone called erythropoietin, or EPO, telling your bone marrow to make more red blood cells because you want to get more blood volume so your blood pressure goes back up. How about blood pH? Well, your kidneys and your lungs are the two systems the two organs that maintain your body's pH. If you need to get rid of more acids, well, you're going to pee out more hydrogen ions. If you need to conserve more acid, you're going to actually reabsorb hydrogen ions instead. So your kidneys do play a role in maintaining your pH. What is the technical term for urination? It is micturition. M-I-C-T-U-R-I-T-I-O-N. Micturition. So the urination is actually caused by the micturition reflex. A couple more minor ones. What organ does the urinary bladder resemble? It's actually the stomach. They both have, every other hollow organ but those two has 2 layers of smooth muscle. The stomach and the bladder have a 3rd layer of muscle. They also have folds inside of them called rugae, R-U-G-A-E, that allows those 2 organs to expand and relax. So your stomach and bladder can both be empty and they both can be very full. So they actually really, really similar structurally. Okay, lastly, what makes alcohol such a powerful diuretic? Alcohol actually stops the release of antidiuretic hormone. And like I mentioned earlier, if you don't have ADH, you're producing 27 liters of urine a day, which we can't allow. So alcohol is a very powerful diuretic for that reason. All right, good luck.